On the 29th of April, 2016, the American Academy of Orthopedic Surgeons published an article on embryonic stem cells and their potential for spinal cord injury recovery.
Although there is no author credited, it is clear a lot of people from the Academy worked on this article as it goes through the potential of stem cell therapy, where the source of these stem cells could come from. Embryo stem cells were discovered on the 9th of November 1998 by a team at the University of Wisconsin-Madison led by Jeffrey Jones and James Thomas.
There is a lot to unpack before we go to the heart of this topic. First of all, what are stem cells, and why are embryonic stem cells so significant? Stem cells are cells that have the ability to develop into many certain functional cells of the human body. Cell differentiation is the process of which this stem cell divides into many functional cells. Embryonic stem cells are stem cells collected from an embryo. What makes these stem cells so significant is their property of pluripotency. Pluripotency means the cell can become any cell of the body which is functional. Adult stem cells, on the other hand, are only multipotent, which means they are limited to which cells they can become depending on when and where it is needed.
The embryonic stem cells are collected during the blastocyst stage of an embryo's development, which is typically around 4 to 6 days after fertilization. Stem cell therapy has been done in combination with gene therapy, where the insertion of genes is done to fix problems that are mainly inherited and unfixable by common medicine.
Here is a video about the difference between adult stem cells and embryonic stem cells.
Hey guys, welcome back. For this week's video, I will be distinguishing between embryonic stem cells and adult stem cells. So embryonic stem cells are primitive, undifferentiated cells that are derived from a 5-day pre-implantation embryo. So this is an embryo that has been fertilized outside the uterus. For example, in an IVF clinic that are left over or no longer needed can be donated to research with the consent of the donors or parents. So these cells are capable of dividing without differentiating for a long period of time in culture and often stimulate to differentiate, so change into a specialized cell. This is done by a scientist changing the chemical composition of the culture in which they are present, or altering the surface of the culture dish, or modifying the cells by inserting specific genes. So ultimately these cells have the potential to be anything, which means they're pluripotent, which means they are able to give rise to all the various types of cells within the body. Adult stem cells are also known as somatic stem cells, and these stem cells are also undifferentiated but are found in the body after development that multiply through cell division in order to replenish dying cells and regenerate damaged tissues in adults. These cells can be found in bone marrow but are so rare, only about 1 in 10,000 to 15,000 cells in bone marrow are stem cells. So here is a table outlining the main differences between the two types of discussed stem cells. So embryonic stem cells are pluripotent, and they can become all cell types of the body because they are pluripotent. They can be grown relatively easily in culture, but scientists believe that tissue derived from embryonic stem cells may increase the likelihood of being rejected after transplantation, but it is unknown as very few clinical trials have tested this. These cells are also considered ethically controversial. Adult stem cells, however, are multipotent stem cells, so they have the capacity to self-renew by dividing and to develop into multiple specialized cells in a specific tissue or organ. However, they are limited to differentiating into different cell types of their tissue of origin, and are also rare in mature tissues, so isolating these cells from an adult tissue is challenging, and methods to expand their numbers in cell culture, so growth of cells in vitro in an artificial medium for research or medical treatment, have not yet been worked out. So this is important as a large number of cells are needed for stem cell replacement therapies. However, adult stem cells and tissues derived from them are currently believed less likely to initiate rejection after transplantation. This is because the patient's own cells could be expanded in culture, coaxed into assuming a specific cell type, so differentiation, and then reintroduced into the patient. So a patient's own cells means that the cells are less likely to be rejected by the immune system, and this is so beneficial because immunosuppressant drugs have awful side effects, and they also are considered more ethically acceptable.
The genetics involved in this topic is rather vast, as stem cells often aim to fix genotypic diseases like spinal cord injuries and cancers. In some cases, people have had a nucleotide base within their DNA deleted, leading to life-threatening conditions. In combination with gene therapy, the insertion of stem cell in genes has ways to treat people with this certain genetic mutation.
Intravenous infusions in the bone marrow of a person has shown the ability to treat genetic diseases like leukemia, bone marrow aplasia, and congenital immune deficiencies.
The significance in this discovery is major, since it has the potential to save many lives and improve upon the medicines we currently have for treating spinal cord injuries. It is also a good place to find pluripotent stem cells. This will be able to treat spinal cord injuries, cancers, and diabetes type 1. The implementation of this in combination with gene therapy has also shown many promising results and a possible future for treatment of spinal cord injuries. Researchers within the biomarker will also be able to develop new prescription drugs for people suffering from this condition.
The limitations are, however, that stem cell therapy can only be taken on people above the age of 18, as well as various bioethical concerns encompassing embryonic stem cell therapy. In most circumstances, embryos are treated as a human creature, meaning it should have the rights of a human, and killing embryos off can be considered along the lines of abortion, which is becoming more legal in areas of the world but still, however, unethical in most circumstances.
The topic of embryonic stem cells for spinal injuries is still an under-researched topic and is hard to find further research due to the ethical concerns. Are there any other good sources of pluripotent cells to replace the need for embryonic cells, and is there a way to extract embryonic cells without killing embryos? Can stem cell therapy lead us onto a future we are actually more worse off of? These are some of the many various questions that are yet to be answered, but hoping to be researched in the near future.
Hopefully you have learnt more about the process of embryonic stem cells for spinal injuries and are able to understand the bioethical problems surrounding this under-researched topic. Thank you for listening.